Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Imagine a molecular editor so precise, so versatile, you could reprogram it with just, well, a simple genetic instruction. Forget complex protein redesigns, think plug and play. This wasn't just a dream back in 2012. Today, we're diving into the discovery that fundamentally changed how we think about editing the very blueprint of life, essentially, democratizing genome editing almost overnight.
Indeed. Our deep dive focuses on this really groundbreaking 2012 science paper by Jinnick and colleagues. And the true aha moment here wasn't just how the Cas9 protein worked with a sophisticated 2-RNA guidance system to precisely cut double-stranded DNA. It was realizing that this intricate natural system could actually be boiled down to a single, easily engineered RNA molecule.
Wow.
Yeah. This immediately transformed a biological curiosity into a plug-and-play molecular tool, opening the door for routine genome editing in a way that, frankly, had previously been unimaginable.
So, to set the stage for you, our listener, what did we actually know about CRISPR before this paper dropped in 2012? It kind of felt like something whispered about in labs, maybe a cool biological quirk, but not quite the powerhouse it became later.
Well, by then, CRISPR systems were known as a fascinating adaptive immune system in bacteria and archaea. Think of it as these microscopic organisms keeping a sort of genetic rogues gallery. They'd remember invading DNA from viruses or plasmids by storing short spacer sequences, almost like genetic mugshots, you could say.
Huh, like little memories.
Exactly. These spacers would then be transcribed into guide RNAs to direct a defense mechanism against future attacks. It was an incredible bacterial immune memory system, but the how it actually worked biochemically was still pretty murky.
So they had this amazing genetic memory, but for type 2 systems, the ones involving the Cas9 protein specifically, what was the big puzzle then? What stopped scientists from turning this curiosity into, a practical tool?
Precisely. While genetic studies had implicated two specific RNAs, the CRISPR RNA, or EQR RNA, and a transactivating cRNA known as TREC RNA, the exact mechanism. How Cas9, directed by these RNAs, biochemically cleaves specific DNA targets. That was still a black box. What really stumped researchers was the lack of a clean in vitro demonstration. There wasn't a simple recipe linking an RNA sequence to a programmable nuclease that would only cut when a specific feature called the protospacer adjacent motif, or PAM, was present.
Right.
The field urgently needed to convert this genetic curiosity into a definable molecular machine to truly unlock its potential. I mean, for anyone who had wrestled with the complexities of designing custom proteins for ZFNs or TALENs back then,
Oh yeah, that was tough stuff.
right, the idea of simply typing in a new RNA sequence to reprogram a gene editor was almost unbelievable if, and it was big if, it could ever be made to work reliably.
Okay, let's unpack this. How did the Jinnick team actually start to unravel the mystery of Cas9's cutting ability? What were those first experimental steps in turning that genetic curiosity into a molecular machine scientists could actually, work with?
They started with what we call a minimal reconstitution. Basically, they purified the Cas9 protein from Streptococcus pyogenes, a common bacterium, and set up carefully controlled experiments outside of a living cell, in a test tube, essentially.
Okay, in vitro.
Exactly. And crucially, they showed that mature cRNA alone couldn't direct Cas9 to cleave target plasmid DNA. It just didn't work.
Right.
However, the dramatic moment came with the addition of the tRNA RNA. This other RNA forms a base-paired structure with the cRNA, and this pairing suddenly triggered Cas9 to cleave the DNA. This reaction specifically required both magnesium ions, which are often needed for enzymes like this, and critically, a cRNA sequence that was complementary to the target DNA sequence.
So the cRNA wasn't enough on its own. It really needed the TRAC RNA as a partner, almost like a co-pilot activating the main engine, maybe?
That's a great analogy, yeah. And how precise was this cutting? Were we talking about a rough chop or something more surgical?
Yeah, exactly, like a targeted laser.
Extremely precise. They found that this dual RNA-guided Cas9 introduced double-stranded breaks at very specific sites. the DNA strand complementary to the key RNA guide sequence. That was cleaved exactly 3 base pairs upstream of the PAM sequence.
Okay.
The non-complementary strand, the other one, was cleaved at one or maybe a couple of sites, but generally between 3 to 8 base pairs upstream of the PAM.
So it's like cutting a specific length of fabric, but then snipping the other side at a slightly offset point, creating these sticky ends or an overhang that's really crucial for how cells try to repair the break later on.
Exactly. This established a critical biochemical sufficiency, meaning these components were enough, and a simple targeting rule. Remarkably, it was behaving like a sequence-specific restriction enzyme, which biologists used all the time.
Right, but those are proteins recognizing DNA directly.
Exactly. This one was guided by RNA, which is much easier to change.
Fascinating. So Cas9 cuts both strands of DNA. Does it use 2 separate scissors for that, or just one really clever one that manages to do both cuts?
That's a great question, and the team investigated exactly that. Cas9, the protein itself, contains 2 distinct nucleus domains. Think of them as the cutting parts. One is called an H&H domain, and the other is a Rho-VC-like domain.
Okay.
So what they did was clever. They created Cas9 variants, mutants, with inactivating point mutations in one or the other of these domains. This way, they could see what happened if only one scissor was working.
And what did they find with these mutant Cas9 proteins? Was it a single blade operation, or did they indeed have 2 distinct cutting edges working together?
Well, when Cas9 had only one active nucleus domain, say just the H&H or just the Rho-VC, it produced nicked open circular plasmids.
Ah, meaning only one strand of the DNA was cut, like a nick, not a full break.
Precisely, just a single strand cut.
Yeah.
In stark contrast, the wild-type Cas9, the normal one with both domains active, produced a linear DNA product.
Indicating a complete double-stranded break. Got it.
Yeah, exactly. Further work then precisely mapped which domain cut which strand. It turns out the H&H domain cleaves the complementary DNA strand, the one the RNA binds to. And the Rho-VC-like domain cleaves the non-complementary DNA strand, the other one.
That's incredibly elegant, like a molecular 2-person cutting team, each specialized for a specific task, working together perfectly to achieve that complete break.
It really is. And the double-stranded break, that's the crucial cellular distress signal. That's what the cell tries to repair, and that's the point where we can introduce the desired genetic changes in genome editing.
Right, that break is key. So that's incredibly elegant, 2 specialized domains, each tackling one strand. But okay, once the cutting machinery is understood, how does this dual RNA complex actually find its target DNA? What are the rules for recognition? How does it home in on the right spot in that huge genetic landscape and not just cut anywhere?
Yeah, that's critical. And the key insight here was that the trachRNA isn't just for cleavage. It's also absolutely crucial for DNA binding.
Really? For binding too?
Yes. Cas9 alone, or even Cas9 with just the cRNA, showed very little specific binding to the target DNA in their tests. But add the trachRNA, and suddenly, target DNA binding was substantially enhanced. This suggests that trachRNA helps properly orient the cRNA for interaction, acting almost like an anchor or maybe a scaffold that helps the entire complex latch onto the DNA correctly.
So it's not just a cutting cofactor, it's
No.
a crucial targeting component, a kind of molecular grappling hook.
Okay.
And what about the guide RNA sequence itself? Are there any hot spots for binding? Like, is there a specific segment of the RNA that absolutely has to match the DNA for it to work well?
Yes, their studies revealed a critical seed region. They found that mutations in the part of the guide RNA region that binds closest to the PAM sequence, those were poorly tolerated.
Ah.
Making changes there significantly decreased DNA cleavage efficiency. This seed region is located at the 3' end of the protospacer sequence in the DNA target. It needs at least 13 contiguous base pairs of complementarity between the cRNA and the target DNA for efficient cleavage.
13 base pairs. Okay, that's pretty specific.
It is. But interestingly, the system could tolerate up to 6 contiguous mismatches further away in the 5' terminal region of the cRNA. So it really highlights the seed region's outsized importance in initiating that stable binding.
Okay, so the seed region near the PAM is key, and we can't forget about that mysterious PAM sequence itself. What role does that play in all of this? Is it just a bystander or maybe something more active, like a molecular permission slip?
The PAM, the protospacer adjacent motif, is absolutely essential. For the S. pyogenes system they studied, the PAM conforms to an NG consensus sequence, meaning any nucleotide followed by 2 guanines.
NGJ, okay.
And it's located just 1 base pair downstream of where the cRNA actually binds in the target DNA. Their experiments clearly show that mutating this GG motif, especially on the non-complementary strand of the DNA, dramatically reduced both Cas9 catalyzed cleavage and the binding affinity of the Cas9 RNA complex for its target DNA.
So the PAM isn't just a label. It's like the designated doorbell that Cas9 has to ring to get permission to even unwind the DNA and, effectively peel open the strands, forming that R-loop structure before it even considers cutting.
That's a fantastic way to put it. It's not just a bystander. It's a gatekeeper, exactly. A kind of molecular password for Cas9 to even engage with the DNA. That's a much more dynamic role than just being a simple recognition site, isn't it?
Yeah, it really is. It's almost like a go signal or maybe permission to proceed signal.
Exactly. It's thought to be required to license duplex unwinding, strand invasion, and the formation of that R-loop structure. Essentially, the PAM determines if Cas9 should even consider binding and cutting at a particular spot. It's a critical part of the targeting specificity, making sure it doesn't just slice DNA indiscriminately.
Okay, so knowing these precise rules for targeting the seed region, the absolute need for the PAM that naturally led the team to ask, could this complex 2-RNA system be simplified? This dual RNA system, with all its precise rules and components, it sounds incredibly sophisticated, really elegant biology. But for a scientist in a lab, having 2 separate RNA molecules to make and handle for every single experiment, that sounds a bit cumbersome, right? A bit fiddly.
It definitely adds complexity, yeah.
So that's where things get really interesting for practical application. Was there a way to simplify this elegant biological machinery for easier use? And just playing devil's advocate here, wouldn't a single, much longer RNA molecule maybe be more prone to degradation inside a cell, or perhaps introduce new folding problems compared to 2 separate, maybe more stable pieces?
That's a very fair point, and it was certainly a risk they considered. But the team had this brilliant insight. If the cRNA and trachRNA naturally formed a stable, base-paired structure together, could they just fuse them? Could they link them into a single contiguous RNA molecule?
Yeah, just stitch them together.
Essentially, yes. This was the pivotal engineering leap, creating what we now call a single-guide RNA, or sgRNA. It was a true moment of ingenuity, taking this complex natural system and figuring out how to make it much more user-friendly, despite those potential challenges you mentioned, like stability or folding.
A single chimeric RNA. That sounds like a stroke of genius, honestly. But did it actually work as effectively as the 2 separate RNAs? Or did that simplification come at a cost? Maybe less efficiency or specificity?
Remarkably, it worked beautifully. They designed and tested different versions of this chimeric RNA, effectively linking the 3' end of the cRNA to the 5' end of the trachRNA with a short linker sequence, often something simple like a tetraloop.
Okay.
And this single RNA molecule successfully guided Cas9 to cleave target DNA just as effectively, just as specifically as the 2 separate RNAs did. It really proved it could overcome those potential practical issues. And they even demonstrated its versatility. They designed 5 different sgRNAs to target a common lab tool, a gene for green fluorescent protein, GFP. And they showed efficient, correct cleavage in all 5 cases. This proved the concept was robust and broadly applicable.
That is amazing.
I remember when this paper came out, there was this buzz. You could feel it in the labs, almost a collective gasp of, wow, why didn't we think of that earlier? It was just so elegant in its simplicity.
That's a huge step. So what does this all mean for the bigger picture? That plug-and-play concept you mentioned, the idea that you just swap out an RNA guide instead of redesigning a whole protein, how profoundly did that actually change the pace and the accessibility of genome editing in labs around the world?
Oh, it was a true paradigm shift. The invention of the single-guide RNA meant that, really, for the first time, researchers could program a DNA endonuclease, Cas9, to target virtually any desired DNA sequence by simply changing a short RNA sequence.
Just the guide part.
Just the guide part. This was a monumental shift compared to the previous tools like zinc finger nucleases, zidafens, and talons. Those required engineering a completely new, complex protein for each target site.
Which took ages, right, and a lot of expertise.
Exactly. It was far more laborious, time-consuming, and frankly, expensive. This sgRNA discovery slashed the conceptual cost of retargeting, as one analysis nicely put it. It effectively democratized the ability to edit genes.
It's like switching from, blacksmithing every new custom tool you need to just downloading an app or maybe 3D printing a new part using just a simple text file instruction. It transformed genome editing from this bespoke craftsmanship into rapid, information-driven design.
That's a perfect analogy.
Exactly.
If we connect this to the bigger picture, this discovery converted CRISPR-Cas9 from a somewhat mysterious black box bacterial defense mechanism into a rules-based molecular machine. It provided the essential abstraction, the simplification, that enabled its immediate extension into living cells and whole organisms by labs everywhere. It's arguably the root citation, the foundational paper, for nearly every Cas9 application we see today, from large-scale functional genomic screens to all the therapeutic development happening now. And it really laid the foundation for future innovations built upon it, like NICUS's, base editors, prime editors, all those came later, building on this core idea.
Right.
It truly ignited the modern era of CRISPR genome engineering.
What really stands out to me listening to this is that this paper was more than just a detailed biological finding. It was a conceptual breakthrough. It streamlined genome editing, making it accessible and programmable in a way that was just unimaginable before. It was a leap from understanding a complex natural system to brilliantly simplifying it for human ingenuity.
Absolutely. This deep dive into the genetic paper really shows us how understanding the fundamental mechanisms of a natural system down to the biochemical details can unlock its potential for incredible technological applications. It clarified how bacteria's sequence memory could be translated into targeted action, into precise cutting, and in doing so, it offered humanity this unprecedented tool that honestly we are still just beginning to fully explore and apply responsibly.
Think about it. From a humble bacterial immune system, something evolved over millennia for defense, to a tool that could potentially rewrite the very code of life just by changing a short piece of RNA. It makes you wonder, doesn't it? What other elegant natural systems are out there, waiting for us to uncover their programmable secrets? This paper, even though it was initially an in vitro biochemical study done in a test tube, it opened up entire new research fronts. Everything from how do we safely deliver these tools into cells, to how do we precisely control the DNA repair pathways afterwards, and crucially, how do we mitigate any off-target effects? It's a real testament to how foundational discoveries continue to inspire decades of innovation, constantly pushing the boundaries of what's possible in biology and medicine.